Good afternoon, everyone, and welcome to the 2016 Joseph J. Kinyoun Memorial Lecture. NIAID has hosted, could I have the next slide? NIAID has hosted this lecture series since 1979 in honor of Dr. Joseph Kinyoun, who some of you may not know was a pioneer of modern biomedical research, who founded the Hygienic Laboratory in 1887. And his work ushered in what we now know and realize was a new era in America of combating infectious diseases through the rigid scientific research process. And since then, his original one-room laboratory, which was housed in the attic of the Staten Island Marine Hospital in New York Harbor, has evolved into the 27 institutes and centers, which we now know today as the NIH. His vision continues to inspire our approach to improving human health. Next slide. It is my pleasure to introduce today's speaker, Dr. John Mascola. John, as many of you know, is the director of the NIAID's Dale and Betty Bumpers Vaccine Research Center and an internationally recognized leader in HIV vaccine research who has made many seminal contributions to the field. At the VRC, John provides overall direction and scientific leadership for the basic clinical and translational research activities aimed at developing and testing candidate vaccines not only against HIV, but other diseases such as influenza, Ebola, chikungunya, Zika, and other issues of major global health consequence. He also serves as one of my principal advisors on vaccines and related biomedical research affairs. And additionally, he is the chief of the virology laboratory at the VRC, where he directs research on structure-based design and testing of HIV and influenza vaccines and on optimizing the immune responses to and identifying the correlates of immune protection for these vaccines. In 1996, before John joined the VRC, he was one of a group of investigators who discovered that easy-to-induce HIV antibodies neutralized only laboratory-adapted viruses and not those obtained from HIV-infected individuals. And a few years later, he began a truly pivotal series of experiments on the biology of HIV-neutralizing antibodies and was among the first to demonstrate that passively administered antibodies could protect non-human primates challenged with the simian form of HIV. In what is now a classic 2007 paper in Nature Medicine, he was the first to show that CD4 binding site antibodies isolated from the plasma of HIV-infected individuals could neutralize diverse strains of HIV, and this led to further research culminating in the seminal 2010 paper that he co-authored in the journal Science describing VRC-01, the potent CD4 binding site antibody that neutralizes more than 90% of HIV strains. He then went on to lead to the development of VRC-01 for preventive and therapeutic use, ultimately achieving a good manufacturing practice production of the antibody and launching currently ongoing human clinical trials to study it. In recent years, John has also led efforts to map the co-evolution of HIV with broadly neutralizing antibody and has isolated such antibodies in this setting. John is not only a visionary and highly productive scientist, but also a wise and generous person from whom countless researchers seek guidance, including myself. Please join me in welcoming John Mascola.
So good afternoon, everybody, and thank you, Dr. Fauci, for the kind introduction. I'm honored to be asked to give this Kinyoun lecture this year, and actually quite excited about a topic which I discussed with Tony. And the idea of the talk today will be to give you all some sense of why, for some vaccines, for some diseases for which we don't yet have a vaccine, we need to apply these tools that we call structure-based vaccine design and B-cell ontogeny. And so the way I'll structure the talk is to first give you some background on vaccines in general, the successes but also remaining challenges for vaccines. And then I'm going to explain these terms because I appreciate that B-cell ontogeny is not an inherently obvious term to everybody. And And to give you examples of how we use these tools, I chose 3 viruses. So the talk will focus mainly on viruses, but the viruses will be RSV, HIV, and influenza. So first, let's talk a little bit about the public health impact of vaccines. And these are data from the Centers for Disease Control that are compiled every few years. The red bar shows the number of cases in a year of any given disease in the pre-vaccine era, and the blue bar shows the year 2012. And you recognize diseases, measles, mumps, hepatitis A, and note that there were hundreds of thousands of cases per year in the pre-vaccine era, and then there are now hundreds or no cases at all. And one can actually look at the actual numbers, which I think is helpful. I kind of like or use the example of measles, mumps, rubella, because I think everybody knows that their children get that at age 15 months, and students get the booster before they go to college. And you see that in the 1950s, hundreds of thousands of cases going down to essentially a handful of these cases. Congenital rubella syndrome no longer happens in the United States, nor does paralytic polio, all because of the success of vaccines. So how did we get there? We used what we now call classic vaccine approaches. We can grow up a virus, and again, I'm using viruses as an example, but bacterial vaccines often use similar conceptual vaccine technologies. We can live attenuate the virus. We can kill it chemically. And then in more recent years, so in 1986, we had our first recombinant protein subunit vaccine, which was hepatitis B. And in the 2000s, we had our first virus-like particle vaccine, which is an empty particle with no nucleic acid, and that was based on pioneering work of Doug Lowy and John Stiller here at NCI NIH. So now we call all of these pretty classic approaches, and all of them stimulate the body's immune system to generate both antibodies, and I'll focus more on the antibody response here, but also importantly, helper T cells and killer T cells. Good vaccines give us lifelong protective immunity at a pretty high level. Importantly, though, there are many human diseases for which we don't have a vaccine. So I list some of them, not all of them here, but we often talk about the big 3 being malaria, tuberculosis, and HIV because these diseases cause such a huge global epidemic. Respiratory syncytial virus, cytomegalovirus, Epstein-Barr virus, hepatitis C, chikungunya, all diseases for which we don't have a vaccine. Many parasitic diseases that I won't talk about also are in great need of effective vaccines. If we look again at CDC data for just the United States, you can see the number of cases for these, and oftentimes people don't think about hepatitis C or norovirus. Millions of cases for some of these, even if it's just bad gastroenteritis or a respiratory infection, with sometimes thousands of deaths. Even influenza, for which we have what you really can say is just a modestly effective vaccine, tens of thousands of cases a year. I particularly like a little spot on the CNN website that someone brought to my attention because it was during the 2014 Ebola outbreak, where I think everybody remembers that we had very concerning imported cases, some treated here at the clinical center. But this little spot brought up the, I think, reasonable point that if you think about the scariest viruses, even in the United States, it may not be Ebola. It's all of these where there are many, many cases. It was a nice educational piece, I thought. But let's look outside the United States. So according to data that are compiled annually by the World Health Organization, infectious diseases are the second leading cause of death and disability in the world, second only to heart disease and stroke. And we go back to these major disease entities, TB, HIV, malaria, respiratory diseases in general, diarrheal diseases in general, cause on the order of millions of deaths globally a year, disproportionately affecting children oftentimes. And in all of these cases, vaccine development could have a major impact. And then lastly, we keep learning about new diseases. We think of Ebola as an old disease, but that was really recognized only in 1976. HIV only recognized in 1981. SARS, chikungunya, MERS, Zika, and you can be assured that there'll be a disease here down the line in another few years. So the need for vaccines persists. And so we have many successes, but we have obvious, clear viruses and other pathogens for which we don't have a vaccine. The question is why, and what can we do about it? So I'm going to explain what I mean by structure-based vaccine design and B-cell ontogeny and give you some examples. So by way of definitions, there are no dictionary definitions for these because they're pretty new concepts. So I made these up, and I take full responsibility for any imprecision in them. But for structure-based vaccine design, what we generally mean, and I'll use a virus as an example just to make it a bit more tangible, we use atomic level structural information to manipulate or change a viral protein to improve its antigenic profile and make it a better vaccine. And then B-cell ontogeny was really coined based on the classical definition of ontogeny that we know in biology, which is the developmental history of an organism from a fertilized egg to a mature organism. So we borrow that concept and say that starting from a single B cell, which produces antibodies, we study the evolution of an antibody response, which we call an antibody family or an antibody clonal family, as it evolves through the process of affinity maturation. So those are the definitions. It may be a little bit easier to look at it visually, and many people know HIV here in the room. So we know that HIV has a surface envelope glycoprotein, the envelope. We actually, and I'll talk about this in a minute, we have the structure of that envelope, and we have antibodies that bind to that envelope. So you can think about structure-based vaccine design as saying, given a known antibody that we think is a good antibody, and given its known viral epitope here, can we design a vaccine to elicit that antibody? If we know the structure, we should be able to do that. That's structure-based vaccine design. B-cell ontogeny asks, how did the immune system generate this antibody? What was the evolution of the B-cell response? And the point I'll make is that for some diseases, HIV and RSV, we need to know this kind of information, or we likely need to know it to make an effective vaccine. So one other point about structure-based vaccine design and B-cell ontogeny is that they're fairly new concepts. They've been around for a little bit more than 5 years, but really the field is focused on them. So these are pretty new concepts. It's the work of several groups, and I'll highlight those who have contributed, but Duke University and Scripps Research Institute are major groups that have been studying these type of things along with the Vaccine Research Center and investigators at NIAID. I think that these concepts have been strongly influenced by HIV, but certainly now influenza and RSV, the fields have begun to use these concepts. And all of this is still early in the proof of principle stage. So the first vaccines based on structure-based vaccine design are just moving into the clinic. So I'll start with a couple of examples. So RSV to start with, which is probably the most straightforward example we have of structure-based vaccine design. An important point for viral immunology is that despite the fact that the immune system is complex, we need helper T cells, CD8 T cells to control infection, a critical component of all successful viral vaccines, and most vaccines in general, is that we need strong antibody responses. We call them neutralizing antibodies. We have a very good vaccine for hepatitis B, and the antibodies to hepatitis B surface antigen are critical. Our flu vaccines induce antibodies to influenza. They could be better antibodies. And for HIV, we seek to induce antibodies that neutralize HIV. So I'll focus to a large extent on antibodies to the viral surface protein, which you can see here. And I'll start with respiratory syncytial virus. So this virus causes severe pneumonia, particularly in very young children and older adults. Each year, tens of thousands of hospitalizations and thousands of deaths are caused by RSV. Worldwide, this virus kills more children, young children, than any other pathogen except malaria, which is a major killer of children. And we have been working on a vaccine since the 1960s without success. So we know a lot about viruses. We think we're pretty good with vaccines, but we haven't been able to make a vaccine for RSV. And the question is, why is that? And you can read good reviews by Barney Graham and others on the topic. So the vaccine that was tried in the 1960s is a fascinating story. It made perfectly good sense. We had successes with polio and other viruses to make a formalin-inactivated vaccine, grow the virus in the lab, chemically kill it, and that was done. It was made as a vaccine. It was tested in the early 1960s. The vaccine turned out to induce a pretty weak antibody response, and we'll talk about that in a minute. Importantly, the vaccine failed to work. It did not protect children. There was even cases of enhanced pneumonia in children, and that's a pretty complicated story that I won't talk about in detail. But importantly, clearly the vaccine didn't work. So despite more than 50 years of research, we don't have a vaccine for RSV. So I borrowed some slides from really the campus experts on RSV and related respiratory viruses, Peter Collins at LID, Barney Graham at the VRC, to show you that viruses have surface proteins, and a key surface protein on RSV is called the fusion protein, or what we call the F protein. And so I'm going to talk about this F protein a little bit. And so we can look at surface proteins of many viruses. And what's fascinating is that all of these surface proteins actually are related. They're all trimeric proteins. They're all what we call type 1 fusion proteins. So they mediate fusion. And even though the viruses, parainfluenza virus, influenza, HIV, Ebola, are quite different, there are certain structural similarities in all these surface proteins, and they all mediate viral entry into the cell. So how does that happen? Here's the viral fusion protein. It could be any virus, but let's take respiratory syncytial virus. The fusion protein has to contact the cellular receptor. And of course, the virus wants to get into the cell. So first, the protein engages the receptor. Then the protein changes its conformation and inserts a fusion peptide into the cell surface, and it pulls the virus and the host cell together and injects the viral cell fusion. So structural biologists call fusion proteins entry machines because they actually have a thermodynamic entry mechanism where they change conformation and pull the virus and the host cell together. And you can see that here again. And the critical components of entry machine fusion proteins are that they can be metastable. So fusion proteins are designed to alter conformation. And thus they are often inherently unstable, and that's often called metastable. So you can see the fusion protein here, and you can see after it's changed conformation and pulled the host cell and the virus together, you have the post-fusion conformation. That becomes very important for respiratory syncytial virus. So respiratory syncytial virus investigators for years have known that if you look at an electron micrograph of RSV budding from a cell, and you look very closely, you can see two forms of the fusion protein. One form turns out to be pre-fusion, and the other post-fusion. And so investigators have color-coded that here, pre-fusion in red and post-fusion in blue. And you would ask, well, why does the virus have a bunch of post-fusion on its cell surface? That's not what it needs to enter the cell. It needs the pre-fusion active form. And part of the reason is that it's a metastable protein, and depending on the virus, some of the protein triggers into the post-fusion conformation. So you can see that depicted here, where over time, with a temperature or leaving the virus in place at 37 degrees or in a low salt preparation, many of the pre-fusion proteins will become and trigger to be post-fusion. This is work done by Barney Graham with the VRC. And so in retrospect, one of the key explanations related to the vaccine that didn't work is that the pre-F protein, the pre-fusion protein, flips to the post-fusion during the process of inactivation, which was 37 degrees centigrade and a low dose of formaldehyde, which was used to inactivate the virus. So it's very likely that the vaccine preparation looks like this. Most of the protein on the surface in the post-fusion form. So again, this vaccine was tested in the 60s. It induced a weak response, and it didn't work. So what do we understand about that now? So what we think, and the hypothesis from all of that work, was that the vaccine that we would like to have given should have been a vaccine that had mostly pre-fusion F on it. And that can be done by using a live attenuated vaccine, which is still an active research area, or by using more common recombinant protein technologies now, which are hepatitis B and others have been around for years. We can simply express the recombinant pre-F protein. The problem is it's metastable, and what investigators realized is what we need to do is stabilize this protein. So this is structure-based vaccine design to make a stabilized pre-fusion F RSV protein. So how does that work? So first of all, one needs structural information, and the post-fusion form of the protein was solved as a crystal structure in 2011, and the pre-fusion was solved in 2013. So again, this is pretty recent work. Oftentimes in structural biology, we need an antibody to be linked to or bound to a protein to get good crystals. That's what was done here. This is just an example of a crystal from Peter Kwong's group. And this is the publication by the Graham and Kwong laboratories, in this case led by Jason McClellan, where the title is The Structure of RSV-F Glycoprotein Trimer Bound to a Pre-Fusion Specific Antibody. So we have now the structure of pre-fusion F. And so what can you do with that? We simply want to make a vaccine. We have all this structural information. So the goal is to maintain this and prevent this. And if you know enough about the chemistry, that's possible. So what structural biologists often do when they have a crystal structure, so remember this is a trimeric protein, which means it has 3 identical subunits, which are called protomers, is they use what's called a ribbon diagram or a ribbon representation. So you can pull one of the subunits of this picture here, and you can see what the pre-fusion structure looks like in detail. This is the protein here in the cell membrane, or the viral membrane. This is the protein top. And when the pre-fusion protein triggers to post-fusion, the fusion peptide goes from here to way out here, and it sticks into the cell. So you need this conformational change in order for the virus protein to work. So what we seek to do is to maintain this structure and not allow this. And what investigators did is they looked at this, and I'll give the simplified sort of non-structural biologist view, is if you take these two amino acids and you see what they look like here, they're close together. I labeled this wrong, sorry, this is post-fusion, so ignore that. But in the post-fusion, these two amino acids are very separate. They're in very different space. So instead of keeping these as serines, they were changed to cysteine bonds, and they made an engineered cysteine bond to hold this structure in place. So it basically doesn't allow this to change, doesn't allow this to happen. So that's structure-based vaccine design for RSV. There were other mutations that were used. Cavity-filling mutations are hydrophobic amino acids that help keep this structure in place. And sometimes we put a domain on the protein to hold it together. So there were several mutations that in total, we didn't just want to do this in the structural space, we wanted to make the protein. So these mutations in total allowed us to express a stabilized pre-fusion F protein and keep it stable. So now we have something that can be used potentially as a vaccine. And when that was done, the important data are that this protein is more immunogenic. So several groups have tried RSV vaccines in the older sense, and the protein was post-fusion. So the immunogenicity of post-fusion vaccines is not very good. What was hoped and what clearly was shown in monkeys here in immunogenicity was that a pre-fusion protein is a much better immunogen. So in fact, this type of work is now progressing. This work is just 2013. This type of product and technology has been licensed. And importantly, at the Vaccine Center, we're going to do a phase 1 study in early 2017. The concept of the phase 1 study would be to prove the concept that in humans, this type of protein can in fact boost the immune response. Remember, most people, so in elderly populations, they would be pre-immune. You'd have to induce or boost their immune response. And in infants, presumably this would be a better vaccine than trying to go with a pre-F vaccine. So this was one of the first and clearest examples of using structural biology to make or change a protein and change its antigenic profile and its immunogenic profile. I'll give you an example for HIV where we need to use actually both concepts, both structure-based vaccine design and B-cell ontogeny. And why is that? In part because HIV is a more complicated pathogen from the immune standpoint. So for RSV, we basically had an unstable trimer, and that could be fixed, as I just showed you. But if you look at the protein surface is exposed. There are just a few glycans or sugars on it. The opposite sort of part of the spectrum is HIV, where it's also an unstable trimer, so we would need to stabilize it. It's completely covered in sugars or glycans, and it's very diverse. So the purple plot, more purple is, I think we all know there's a lot of antigenic diversity. So with RSV is pretty simple. The immune system can see it. It just needs to be stabilized. Here we have a much more complex situation. Why is that bad for vaccines? Well, the glycosylation limits the available protein surface. The antibody needs at some point to contact a protein surface, and the glycans essentially get in the way. And the diversity means that an antibody that works against one strain may or may not work against another strain. So antibodies, what we need for HIV are antibodies that are able to penetrate the glycan shield and target conserved epitopes. And this has been a major obstacle to HIV vaccine design. It's not easy in the HIV vaccine space to get the right kind of antibodies. So this is evident just by looking at natural history. That's a point that Dr. Fauci often makes. We learn from the natural history of a pathogen. For HIV, one gets viremia, and very quickly we get antibodies to the virus, and that's how we diagnose the infection. But they're binding antibodies. It takes months to get neutralizing antibodies. If this were RSV or most acute viruses, polio, hepatitis A, these antibodies would come up within weeks. With HIV, they come up with months to a year. That's years. That's your first clue that this type of pathogen is different. The type of antibodies we really want, which are more cross-reactive, can take years to come up. So we have, for HIV, an immunological problem in addition to a structural problem. This translates in the real world of vaccinology because we have tried several types of vaccine candidates in HIV as vaccines, and they have failed to work. So GP120 is a simple recombinant protein. So if this is a trimer, the GP120 is one of the subunits. We have used gene-based vaccines where we put genes of HIV into a genetic vector like an adenovirus, or combined DNA and adenovirus, or we've combined pox virus vectors with proteins. These vaccines failed to work. So essentially, they did not protect in placebo-controlled trials at all. And this vaccine protected at a 31% efficacy, which is not good enough yet. We need to build on that. So importantly, we get poor neutralizing antibody responses in all these vaccines. And by that we mean low potency and limited breadth, limited cross-reactivity. So we have a sense from the antibody perspective why that happens. Again, we immunize with GP120, and the antibodies elicited by GP120 immunization do not effectively bind this trimer. So we get lots of antibodies to GP120, but the surface of 120 is buried on the trimer. So if this were RSV, it wouldn't be a problem. What we want are antibodies that actually bind to the trimer and would neutralize the virus. So the question is, how do we get there? How do we take that immunological step? So first-generation vaccine proteins, the monomeric GP120, induced high binding but poor neutralizing antibodies. Natural infection does induce strain-specific neutralizing antibodies in essentially everybody over time. But the broadly neutralizing antibodies that we want with a vaccine, which cross-react with many strains, are found in the sera of only about 20% of HIV-infected people. We don't understand exactly why that is, but part of it is the chronic nature in HIV and the fact that it evades the immune system. So some years ago, we were working with Mark Connors here at NIAID and studied subjects that were part of NIH clinical trials. So here these are 80 individuals, all on the x-axis, who are HIV infected, and each dot is a virus. And what you can see is that some donors make weak neutralizing antibody responses, and for most viruses, they don't neutralize. And some donors neutralize most viruses at high titers. So this was a first clue. I think it's pretty obvious here that we're interested in these donors. These donors make really potent antibody responses, the kind that we need with a vaccine. So what can we do? We can isolate the antibodies from these individuals. I'll show you how we do that. We can study how they neutralize structurally, and we can ask how the immune system makes them. So we can do the structural biology and the B-cell ontology on these donors. The technology to pull out antibodies has advanced in the last few years. So we can take a protein probe, an HIV probe, mix it with B cells, and we can do cell sorting, and these are HIV-specific B cells. We can take an individual B cell and PCR amplify the heavy and light chain antibody genes. That technology has been routine in the last 5 years. So from a B cell, we can express in the laboratory the antibody made by that B cell receptor. So if we do that, and I'll jump ahead some years now, since 2009 to about 2016, the field has isolated literally hundreds of potent neutralizing antibodies against HIV. And these antibodies define the epitopes on the virus. So each of these blue patches are where an antibody binds. It binds here at a site called V3 glycan. So the antibody gets through the glycan, actually interacts or touches the glycan, and touches this patch. Or it touches a green patch here that we call the V1, V2 region. Or the CD4 binding site, which is where the virus engages CD4. So we understand where these viruses attack, and I can show you a very nice video made by Peter Kwong and Jonathan Stuckey at the VRC, where knowing this information allows you to make very nice, really very specific videos like this showing the antibody in the right size. So this is an antibody binding to one epitope or another epitope or the CD4 binding site. You can see how big antibodies are, and you can, I think, appreciate that at the chemical level, the structural level, we know these interactions at the atomic level, atom for atom, exactly how this antibody is binding to this virus. So I borrowed a slide from Dr. Fauci to make the point that the challenge here is translating, the fundamental challenge, epitopes to vaccines. We know the epitopes. We need a vaccine, a specific way to immunize to induce these antibodies. So how do we do that? We basically need to move away from just recombinant proteins to being able to use the trimeric structure. That's what antibodies bind to, and that's what's required to neutralize. If we're not immunizing with something that has what we now call native structure. So the RSV people call it prefusion. The HIV people call the prefusion structure the native virus protein. But we need things that are native. Otherwise, we're not going to get antibodies that actually recognize the virus. So just since 2013, so again, a lot of this is new biology, investigators have solved the crystal structure of the native glycoprotein envelope. And that's done here, the Wilson and Ward groups. And in fact, this is a stabilized protein. So similar to RSV, really independently, HIV researchers realized that you had to put a cysteine bond between GP120 and 41. And here an antibody is bound to the protein that allows crystal structure, and we can see the crystal structure. And then since 2014, Peter Kwong and other laboratories have given us atomic level structural details of not just the protein, but actually the glycan shield around the protein. So we know exactly what we want to immunize with or what the immune system sees. The problem still lies in the diversity of HIV. If we immunize with a trimer, we get antibodies to that trimer. But these antibodies are too strain specific. They don't neutralize diverse viruses. And of course, that's what we need. So we're still left with a viral diversity problem. So we've crossed the threshold in that we can do structure-based vaccine design. We still have to solve this immunological problem for HIV. So we go back to looking at our very important donors. So we have examples in natural history, in natural infection, of people who make cross-reactive antibodies. These antibodies, in some people, neutralize 90% of strains. So what we want to do is we want to follow that B-cell response, which we call B-cell ontogeny. And we want to follow it from the beginning. So we would like to study an individual from early on in infection through 2 or 3 years. And we can do that. There are some very important clinical cohorts. And I won't go through all the names, but these are often NIH-sponsored or funded cohorts where individuals are followed who are at high risk. They are followed from the time before infection through a time of infection, oftentimes through several years of infection. So what we do is we find an individual, we study that individual, we pull out the antibodies at year 2 or 3, and we find antibodies that are very potent and broad. And we go back and we pull out a lot of antibodies. And so now we're talking about antibodies that are in one family. So an antibody that binds the CD4 binding site that are all related. And we want to study exactly how that first B cell made that antibody population, because that's going to tell us how the immune system did it. So the example of that is here. Here are naive B cells before they've seen antigen. One of the B cells sees HIV when someone becomes infected. And remember now that B cell divides. So there are literally hundreds or thousands of B cells that are all from that are all daughters of the same B cell. They make what we call a family of antibodies. All these antibodies are the same clone, same clonal family. And so we can study this whole family by just pulling out one antibody. Once we pulled out one antibody, we know the genetics, we know the sequence, and we can use PCR to find all of the relatives. So we can find any sequence. And if we have longitudinal samples going back in time, we can study exactly how that antibody went from its first B cell to becoming potent. And that's what was done here in some work that we did with Bart Haynes. And so first we pulled out the antibody. And then what Bart Haynes did is he studied 2 things. He studied first the virus that infected this person. So the beauty of these cohorts are that one has the virus that infected a person. So you can see that early on in infection, a virus is what we call monophyletic. It's very clonal. And the virus diversifies over time. Why does it diversify? Well, it's a very mutable virus, but it's also evading the immune system. And so it turns out that you can show very specifically that as the virus mutates, the antibody response to a specific epitope accommodates and evolves. The virus mutates further, and the antibody mutates further. And as the antibody mutates, it actually focuses in on the more conserved epitope. So this whole process we call antibody-virus coevolution. It's very important because what we can do is we can see that for any given antibody, the antibody mutates. This is the process of in the germinal centers of somatic hypermutation. As the antibody mutates, it gets better and better, and it becomes neutralizing. And we can show that very schematically here. It doesn't help you understand the biology so much, but just schematically, there's transmitted founder virus, and the virus mutates, and the antibody mutates over time. So we know that antibody-virus coevolution happens. What we need to understand it is in detail to use it as a vaccine. And so if we merge the B-cell evolution and the structural biology, we have an understanding of what happens. And this is one example of some complex examples. But if we take an antibody to the CD4 binding site, which is something that we've studied in our lab very heavily, the CD4 binding site is sort of buried in here. Right? That's where the virus engages CD4 on the cell surface. So it has to be somewhat exposed. And you can see there's somewhat of a glycan hole here. So the antibodies that bind the CD4 binding site, Peter Kwong and colleagues have learned over years, that they all approach the virus at a very specific restricted angle. That's the only way in. If an antibody to the CD4 binding site tries to come in at a different angle, that antibody may actually bind CD4 as a protein, but it won't come at the right angle on trimer. So remember also that this protein is somewhat metastable. So during infection, GP120 is shed. So the immune system sees all kinds of things. The immune system sees CD4, and the antibodies to CD4 are around. But that antibody to CD4 sees a real trimer, and it can't get in. But over time, there's a affinity maturation. There's intermediate antibodies and mature antibodies. And the mature antibodies, the ones that we're interested in, have the right angle approach. They accommodate glycans. They find the protein surface, and they neutralize. So the immune system has to go to a lot of work to neutralize HIV, but that's what we need to do with a vaccine. And so you can see that here in what I just showed you. And so we can see in this donor who made very good CD4 binding site antibodies what the virus looked like at the beginning. We can see that the virus mutated a little bit and how the antibody responded. So it gives us something that could be a sequence of vaccination. If we recapitulate this process here with just changing the virus a little bit, we should be able to push the antibody response towards the glycan hole and what we want. This concept is called lineage-based vaccine design because we have to know the lineage of the antibody to get there. It is, frankly, something that's unprecedented in vaccinology. We don't have to do that for RSV. We don't care about lineage-based vaccine design because as soon as you immunize with the stabilized trimer, that whole surface is exposed. And we care about it less for flu, but it's critical for RHV. So the approach for HIV is quite complicated. It starts with neutralizing antibodies from people that bind the stable trimer. It requires structure-based vaccine design, and we've done that. We can stabilize the trimer, and we can make subunits. We know what that trimer looks like in such atomic detail, we can make parts of it that are actually native, that are appropriate. We have examples of B-cell ontogeny, and we are starting to do B-cell lineage design. So the first products for B-cell lineage design are going to the clinic. So that sequence of immunogens that I showed you has been made, and proof of concept studies will be done. So when we talk about antibodies in HIV, we have this whole area of vaccine design, which frankly will take some years. It's pretty obvious that for RSV, we may be onto something that could translate in a few years. And for HIV, all this work is going to take iterative experimental medicine over some years, and is pretty hard to do. So another approach is what we call passive immunization. And passive immunization takes advantage of the fact that we actually have antibodies from HIV-infected people that are very potent. So active immunization means we induce these antibodies. Passive immunization means we would just actually passively infuse antibodies. So a byproduct of all this beautiful structural biology work, where we have all of these antibodies bound to the virus, is that we have antibodies in the lab, and we've started to make them clinically. So there is currently an advanced clinical trial that asks a very specific question. Can a passively infused monoclonal antibody prevent HIV infection in high-risk adults? The study is called the Antibody-Mediated Prevention Trial. This antibody that was isolated at the VRC is administered at 2 different doses every 8 weeks, and it's a pretty large phase 2b clinical trial that is being conducted by NIAID-sponsored clinical networks. One is called the Prevention Trials Network or the Vaccine Trials Network. And first volunteers in African populations were in April, and you can see a volunteer here actually getting infused with an antibody. In the long run, the antibody would be given by a simpler method, a subcutaneous method. And this trial is actively enrolling with over 600 and 200 enrollees and going well. So we hope that, and all going well, that this trial will give us an answer, will give us several answers. Can an antibody block or prevent HIV infection in humans? Everything I've told you about our vaccine design effort presumes that to be true, so it would be nice to experimentally confirm it. It would tell us how much antibody we need, which is also critical for vaccinology. If the antibody level that's needed, if the antibody circulates through the system, gets to the mucosal surface, and we believe from experimental animal models that the amount of antibody actually doesn't necessarily need to be that high as long as the antibody is potent. But we need to know that in a human trial. So it helps us set us a bar for vaccination. So that gives you a sense for where we are with HIV. I'm going to end with an example that's somewhat in the middle of RSV and HIV. So we have influenza vaccines. I think everybody should be in the process of getting their flu vaccine on campus here. And that vaccine is classically an inactivated whole virus vaccine. Or in fact, you might hear the term split virus vaccine. And what we do there, or what manufacturers do, is detergent solubilize the vaccine so that it releases the proteins. And that's an inactivated subunit or split virus. So the hemagglutinin and the neuraminidase are semi-purified, and that's your vaccine. And there's also a live attenuated intranasal vaccine that's more often given to children, isn't recommended this year. But interestingly, all of these vaccines lack durability. So we get immunized every year. So if you think about it, you get polio as a child, and you don't get it again. Measles, mumps, rubella, you get as a child and get boosted as a teenager, and essentially get close to lifelong immunity. Flu we get every year. These vaccines lack breadth, so we change the strains in the vaccine most years. And the efficacy of influenza vaccine is 30 to 60% at best. So clearly, we would like to do better. So let's go back to our principles of structural biology. What do we know about flu hemagglutinin? So interestingly, it's a stable trimer, even though it's related. So it's one of the reasons we probably have a vaccine is that one doesn't have to stabilize it. If you make hemagglutinin, it's stable. But look at the amount of glycosylation. It's not as much as HIV, but key areas of influenza are, in fact, covered by sugars or glycans. And there is diversity, especially in the head region. So there are problems with glycosylation and problems with diversity. And there's this problem with influenza that's called immunodominance and well-studied and original antigenic sin that really I'll kind of depict here. So the hemagglutinin protein, which is one of the major targets for the antibody response, can be categorized into these different subtypes. And I think people hear about H1, H5, H3. So you get an H1N1 vaccine, or et cetera. And you hear about avian flu, which is often H5 or more recently H7. So these are quite diverse subtypes of hemagglutinin. And ideally, we would protect against all of them. But currently, our vaccine is a combination of an H1 and an H3. So if we look at what we know structurally, the hemagglutinin protein is quite diverse in the head region. That's what's depicted by the red, and this is just a nice surface plot by Jeffrey Boyington at the VRC. And importantly, current vaccines induce antibodies mostly to the head. And these antibodies tend to be rather strain-specific. And this is the immunodominance problem, or the immunological problem of influenza. There's a lot of interest. So there's a number of approaches to making better flu vaccines. But one that I'll talk about is focusing immune response on this much more conserved region. If antibodies were elicited here, we wouldn't have to change the vaccine so often. So that's the conserved stem region. So how do we do that? If we go back to structure-based vaccine design, we have the atomic level detail of the entire hemagglutinin protein. This is a ribbon diagram. You can see an alpha helix here, and you can see the receptor binding domain of the head. So in the simplest terms, we just want to chop off the head and leave the stem. So that makes for a very unstable protein. It would simply fall apart in the laboratory. So there are some tricks we can do. We can put a stabilization domain here holding the trimers together. We can put it on a nanoparticle. In this case, it's a ferritin protein, which has the right trimeric orientation to hold the protein. So this is what we call a nanoparticle because it's a protein holding our vaccine antigen on it. So these particles have been made, but importantly, it's quite a complicated process actually, but complex yet very approachable from structure-based vaccine design. I won't go through it. It's published in a paper in Nature and Nature Medicine. But through an iterative approach, one can take the head off, you replace it with a linker, and you can stabilize it and put it on the nanoparticle. So all of that's done through an iterative approach of looking at the structure and making proteins and making sure they're stable. But importantly, is this a good vaccine? Is it a good immunogen? So far we have preclinical animal model data to suggest that it is a good immunogen. So in some work done by Gary Nabel and Barney Graham at the VRC, this particular particle, with just the stem now, so we know that we're making antibodies only to the stem, has been tested in animal models. So the stem is based on H1, but the virus challenge in mouse or ferrets is H5. So it's a pretty diverse challenge. And we see very nice protection in those animal models. And you can see here the stabilized stem vaccine protects mice or ferrets from weight loss. And one can actually passively transfer that protection as an antibody from one mouse to another. And so we know that antibodies to the stem can protect in animal models. There's lots of interest, similar to HIV, in actually making some of these antibodies clinically. So there are clinical trials ongoing by some large pharmaceutical companies to ask if antibodies to the stem can protect or treat influenza. That would be key data. Similar to the HIV trial I mentioned, if these antibodies work to the stem, then that gives us a guidepost to say, well, we need to induce similar antibodies. So currently there are several approaches to influenza. I just showed you this stem-based approach. One can also use nanoparticle technology to put the whole hemagglutinin. So this is the whole protein. So it's somewhat similar to the killed vaccine, but it has some advantages. First of all, we could probably make it more quickly. the stem region may be more exposed. And when you put proteins on a particle, you multimerize the protein, and you get better B-cell receptor stimulation. So potentially, these could be better vaccines. This type of particle will go into phase 1 at our center in 2017, led by Julie Ledgerwood. And then there are earlier stages of research. So yes, the head is variable, but what can we do about variable regions? We know that antibodies to the receptor binding domain are important. If we have nanoparticles, why can't we put H1 and H5 and H3 on one nanoparticle and maybe get a diverse response right off the bat? And so those types of research directions are in progress. So people often talk about a universal flu vaccine, and certainly that's the goal. We think it more from the perspective of how do we improve and make a vaccine better than what we have. So what characteristics are we looking for? Better protection. So like measles, mumps, rubella, like polio, like hepatitis A, hepatitis B, we need to be in the 80 to 90% range. And so some of these technologies could get us there. We need better coverage, which means we need one vaccine that works against prevalent strains so we don't need to change it every year. Think about how transforming that would be if you got your flu vaccine and it protected against strains that are going to emerge 5 years from now, and it was durable. So it's not so far-fetched. We only get booster vaccines once every 10 years for most diseases. So what we want is a highly effective vaccine that you would get once every 10 years. That's the goal. It's approachable with structure-based vaccine design. So I'll summarize the examples I've given you to say that for structure-based design and B-cell ontogeny, these are new concepts in vaccinology. They've emerged over the last 5 years. We still need to generate, and we're beginning to generate, literally in months in the next year, proof of principle in humans. For RSV and flu, structure-based vaccine products are in development with the first clinical trials in the coming year. And they will give us really key data about the way forward for structure-based vaccine design. For HIV and for other diseases like flu that I just mentioned, the monoclonal antibodies that have derived from structure-based vaccine design not only have their place in vaccine design, but also have their place in passive protection. So we have antibodies that are moving into clinical trials, which would never be here if it weren't for the structure-based vaccine design efforts. And then for HIV, I think this is where the first lineage-based vaccine design proof of concept trials will take place, and will begin in 2017. I'd like to end and make some acknowledgments. The concepts of structure-based vaccine design and B-cell ontogeny are obviously in the minds of many investigators. The Division of AIDS has funded 2 centers for vaccine immunology and immunogen design that bring together consortia of investigators, and those investigators have been very instrumental at Scripps and in Duke, and we work very closely with all of them, as well as others, on these concepts of structure-based vaccine design and B-cell ontogeny. And then finally, I'd like to thank the investigators of the Vaccine Research Center, who have all intellectually contributed to the concepts that I've talked about. And then very specifically, Barney Graham and Peter Kwong did much of the work I showed you, and I borrowed heavily from their work and their slides for RSV and influenza. And Barney and Peter will be the first to tell you that the work is done by many of the people in this lab. The RSV work was initially done by Jason McClellan, who worked both with Peter and Barney, and now has his own laboratory and continues to collaborate with us on Ebola and other structure-based vaccine design. And then lastly, too many people to thank, but the laboratories at the VRC that have participated in the HIV part of the work are the Humoral Immunology Lab and the Virology Lab, the Structural Biology Lab, the Bioinformatics Lab, and Mark Carner's lab, who we've worked with for many years in HIV. And then I will stop there. Thank you.
So thank you very much, John, for that extremely elegant lecture. Questions or comments? Hugh, you have a question? Mm-hmm. Good.
John, that was a spectacular talk. Thank you. Do you think the RSV enhancement problem will go away because of the higher titers, or what do you think is going to be the outcome there?
I think it should potentially be solvable in a situation where you have the predominant antibody response to be neutralizing. So with the prior vaccines, the predominant antibody response was weak and non-neutralizing. That left for the possibility of viral aggregates and deposition in lung tissues, together potentially with that also mediating CD8 and sort of abnormal T cell responses. So I think the sense in the field is that if your predominant response is in the right direction, you don't get those off-target immunological responses and should be able to avoid the enhancement problem.
So, John, in the sequential vaccination model of HIV, where you immunize and then sequentially coax the immune response, the question that I always get concerned about is that if you have the initial priming with an immunogen, is the secondary and tertiary and fourth one, how common is that going to be from one individual to another? Or how do you know that the immunogens are going to be inducing the next stage in a common way?
And this is obviously a fundamental question. And when we study a single individual that I showed you, you could wonder, and I hope you did wonder, well, that's one person, right? So is this generalizable? And I would have to say, when I came into it, we studied those individuals with BART. My bias was that it may not be generalizable. But we've learned, and I've changed my view, because what we've learned is that we have these fantastic examples in flu, for example, that responses are often, there are classes of responses that are common among many people. And so I think we use this term B-cell lineage design to say that if we're smart enough about it, for certain categories of antibody, it could be CD4 binding site for HIV, most people can make the response that we're interested in. And if our initial immunogen and our secondary immunogen are designed the right way, they will stimulate a category of antibodies that are common to everybody. It still needs to be proved that it's generalizable, but I think that the B-cell lineage data in now tens of people rather than one person tell us that it should be generalizable in that regard.
For that coaxing along, I assume you want to accelerate it and not take 2 or 3 or 4 years. So can you give a little bit more on what the regimen, the trial design would be?
Yes, and this is also a fundamental problem with HIV. So you saw that nice graph there where it takes several years in HIV. So the question is that in part because the person is infected, the virus infects their CD4 T cells, CD4 help, as Dr. Fauci showed many years ago, the germinal centers are destroyed. And so we hope that in the setting of immunizing a normal, healthy donor, the germinal center response will be normal, and therefore the immune response will be quicker. But also I think there are certain immunological principles like adjuvants and multimerizing the protein and learning how to stimulate a germinal center response that will have to be applied for HIV, because there is a process of affinity maturation where so the antibody in RSV does not have to mature very much. It does not have to mutate very much. You can go through a germinal center reaction and come out and be a memory B cell and be effective. For influenza, it may have to go through several germinal center reactions and mutate a little bit. For HIV, we probably have to promote affinity maturation at a greater level, which means sequential immunization and potentially months of time. Hopefully not years, critically not years, but probably months.
Okay, thank you very much, John.
Thank you.
That was great. That was perfect. That was good. Crystal clear.